All right. Welcome everybody back to this Tuesday section of the RAC Drugs class. I'm happy to have you all with us today. And I'm happy to have Barney King back with us today as well. Before we get started, I will give you our mandatory reminders. I'm sure you guys are all sick of these, but I'm going to do it anyway. So remember to change your screen name to your registered name, just so that we can keep track of who is here for your certificate of attendance. We will be taking a 5-minute break around the top of the hour, so around 7 o'clock, whenever there's a good breaking point, so everyone can have their bio break or get more coffee or whatever you need at that time. Remember to take that survey that will be posted in the chat box later today at towards the end of the session. We do really like that feedback. I do look at all of these, and I do really appreciate getting that feedback. So we use that to make improvements to the sessions, or to keep the sessions the same if you really like them. So again, if there's any questions throughout, keep yourselves mute until you want to ask that question, or you can always type those questions into the chat box and I will ask those to Barney. And so with that, Barney, do you want me, I know we just had you 2 weeks ago. Do you want me to reintroduce you? No, okay.
Well, I've completely changed my biography, but don't let that bother you. So,
all right.
no.
Well, I'll hand it over to you then. Thank you so much.
All right, thank you. I will share my screen and go there, share. And I will, I think, disappear, but maybe. Okay. All right. So hopefully everybody has their speed hearing on. Our plan tonight is that I'm going to talk about the chapter, the European chapter that deals with marketing authorizations for products derived from biotechnology. And then we'll do the biosimilars. My logic for this is that since biosimilar projects are approved via bio pathways, it seems to me to make sense to at least cover that portion. Then we'll come back to the biosimilars. And as we do that, we'll be able to skip at least some of the slides, simply because they'll be somewhat repetitive from this particular group. So with that as the preface, we will move ahead. So first of all, we'll talk about the definitions, what biologic products look like, the legal basis for them. We'll talk a little bit about the development processes, what the marketing approval application looks like, and then post-marketing surveillance. Keep in mind that this chapter specifically addresses the European Union. As we get to the next portion where we talk about biosimilars and we cover the international portion of this, that will of course cover the EU and the US, etc. So we'll come back to some more specifics at that point in time. So biologic medicinal products, the definition more or less is pretty straightforward. It's a medicinal product, the active substance of which is a biological substance. Well, what, you might ask, is a biological substance? It's a substance produced by or extracted from a biological source. And one of the key things about biologics in general is that they need physicochemical biological testing. That enables you to do the characterization and the quality determination, which is, of course, reflective of the production processes and of the controls in place during the course of the processes. So as we talk about biosimilars, those physicochemical biological testing methods and the results from them are really crucial, because that's how you're actually establishing the identity. So biotechnology derived medicinal products, as we work through these definitions, they're all essentially the ones that are in the book. So hopefully by looking at the slides, you can skip having to read at least some of these chapters as you move through the test preparation. Human medicinal products that could act on the body in a similar manner to its naturally occurring biologicals, creating the desired therapeutic effect. So the portions of that definition that say act on the human body, desired therapeutic effect, that's what translates to a medicinal product in the European Union. These are products that are produced from genetically engineered living organisms. The list is there via a biotechnology process. So that process could be recombinant DNA technology, could be use of hybridomas, production of monoclonal antibodies, could be controlled gene expression, etc. So there are several different processes that could be employed to create the products that we're talking about. So the biosimilar medicinal product, and that's what we'll talk about a little bit later tonight, that contains a version of the active substance of biological medicinal product that's then the reference medicinal product that's already authorized in the European Economic Area, which is slightly larger than the European Union. We'll talk about that, and it's actually listed at the bottom of the slide. So the biosimilar product demonstrates similarity to that reference medicinal product based on a comprehensive comparability exercise. And we'll talk a little bit more about that later, but those are the things that are really establishing the quality, the activity, and then ultimately safety and efficacy as you get to clinical trial. So here are examples of biological medicinal products. Remember, we're going to talk about the regulatory processes, and this is true anytime you're looking at the European Union and how it deals with medicinal products. You've got registration procedures, you have types of approvals, and the two things are ultimately related, but they're different sets of categories. And again, we'll come across those as we move through this. So medicines that are derived from the biotechnology processes, you can see the list there. Those go through the centralized procedure. Products that are derived from blood or plasma, you can see the list there. They go through a plasma master file, and the sponsor has an option to go through a centralized procedure, although it's not mandatory. Although, if the product at the end is actually an orphan product, then that would be mandated to go through the centralized procedure. A lot of these kind of products, because of national variability in use and target, frequently go through either the national processes or sometimes mutual recognition, meaning it's already gone through one country and is now recognized in another, or might actually go through a decentralized process. So these types of products actually have multiple ways of going through the authorization process. Most commonly, they're not through the centralized, but in fact, they can be. Things that fit into the category of advanced therapy medicinal products, again, you can see the list on the left-hand side. They do go through the centralized procedure, as well as vaccines, which likewise go through the centralized procedure. So the legal basis, one of the things you'll notice as you read through the books, and it's actually true if you go to the EMA website and you look at various processes for reviewing and looking at drugs and drug development, or for devices for that matter, you'll see shortly after the introduction of this is what we're talking about, there will be a section that will talk about the legal basis for that particular set of processes. So here we'll talk about the legal basis for marketing authorizations. And when we do that, we talk about directives and regulations. And as a reminder, directives are they have a legal basis. They are to be incorporated into the individual national legislation. However, they're to be incorporated, and that means that they, over and above the translation, there can be changes in wording, there can be changes in emphasis of the text that's actually incorporated into the legislation. So as you talk about drug development, for example, the drug directive versus drug regulation, the directives have a lot of leeway to them, whereas in contrast, the regulation has to be verbatim translated to the individual country's legislation or laws. The only variability is the translation, but otherwise the language has to be the same. So the European Union, much as the FDA does, releases guidelines. If you look at the document called the UDRLEX, that's where a lot of this volume 1, volume 2, et cetera, goes. That's a core set of documents that contains the approaches for drugs, biologics, et cetera, and including veterinary medicines, in fact. So that's the first set of legal bases. Those were the drugs. On the biologics. Very similarly, you'll see the directive. And then below that are also the ICH guidelines that are attached to the biologics in particular. So the development process, pretty straightforwardly, you start non-clinically, and particularly with the biologics, you need to be very specifically concerned about the quality processes. I mean, you do in development of all drugs and devices, but here the process becomes key to the success for the project, or the product rather. And so you need to follow all of the quality guidelines. Anything that says ICHQ is applicable in this particular setting. And then from there, you go to the clinical guidelines, and ultimately are concerned with good clinical practices in terms of any of the particular product. So this is what the ICH scheme looks like in general in terms of biologic. On the left-hand side, a part that you don't have as you think about drugs has to do with cell bank. You're from their cell banks, you're developing the processes that ultimately create your drug substance and then your drug product. Then it has to be characterized in various ways so that at the point in time that those data are submitted to the regulatory agency, the agency can, in fact, look at it and say, if this process is adhered to, yes, this product will result. But it's true in the United States, the European inspectors will show up at the plant and they'll watch the process to make sure that what you've described in the piece of paper, well, okay, the electrons today, in terms of the actual submission, that it conforms to what you say you're going to do in actual practice. So once you've done that, you've defined your critical processes, you've defined your specifications, and then at the end, you're ultimately doing your clinical studies. So keep in mind, we're momentarily talking about biologic projects and products in general, not just specifically biosimilars. So here you're looking at your safety, efficacy, risk, and then you're ultimately creating that risk management plan, which is submitted at the time of your marketing approval application. So the format is the format that you recognize well. This is the Common Technical Document format. Just as a reminder, remember that Module 1 is technically not part of the Common Technical Document. It's related to regions. So the US has a version of it. The EU has a version of it. Now the UK has a version of it. Whereas everything below that in sections 2, 3, 4, and 5, those sections are the core of what is called the Common Technical Document. And the organization is basically summaries go to the top, and you get more detail as you go down toward the bottom of that triangle. In terms of the new clinical trial regulation. So clinical trial authorization. As I said, regulations. So that means that this must be transposed into the individual country's regulations verbatim. So the only variation, as I've mentioned previously, really has to do with translation. Other than that, this is what shows up in the individual country's legislative processes. So the key here has to do, again, keeping in mind that we're looking at the biologic products. You've got the dossier. You have one of the advantages, by the way, of the centralized process is you have one application, theoretically get one approval. It applies to the entire European market or to the EEA, so it includes the extra countries. And the review and the whole process has to do with a singular application. So the new clinical trial authorization, that is, what do I have to submit in order to do clinical trial, is set up the same way in parallel. So you submit through the portal in the EU. Again, it's a single document. It's reviewed in a couple different ways in parallel. Processes are set in place in terms of that review and approval. And then ultimately, once you have the approval, you can now sell the product throughout the EU. Keep in mind that reimbursement is determined by individual countries. So yes, you can sell it, but until you get the reimbursement processes put in place, you're giving it away, so to speak. But in any case, so you have a clinical trial authorization process. It's now formally in place. And so this really is the process that enables you to start your clinical development program. These are the type of documents that are required within that clinical trial authorization. It's a table, it's in the book, but I put it here for you for easy reference so you can see what happens. So as I said, two sets of things go in parallel. So you got one that goes to the ethical committees, and then in addition to that, you got it that goes to the individual national competent authorities or the regulators for the respective countries in which you're going to run the trial. The clinical trial authorization process looks like this. You submit, and then there's a validation which says, yes, everything you're supposed to have is in here. That doesn't mean they've approved it. It merely says, yes, all the individual sections are here. Then it's sent off so that now you have the review by the ethical committees and then by the regional, the individual member states. And then from there, the process goes through scientific assessment, through ethics assessment. Again, once these things are started, then they happen in parallel so that the process goes more quickly. Once that's happened, then you can actually initiate your clinical trial. To back backwards a little bit, this is the non-clinical development. I mentioned the ICH guidances. And by and large, in the EMA or the European Union, those are the guidances you're going to follow. So you've got guidances which have to do with preclinical safety evaluation. And just as a reminder, anything that starts with an S really is non-clinical. E, of course, efficacy for clinical, and then the Q for quality, which actually really translates to CMC. You're looking at specific items that have to do with, in this case, a new guideline S12, which having to do with gene therapy. So it's just recently endorsed, so becomes now an official guidance for the EMA. In addition, there are PK and PD studies that are required. Keep in mind, if you're dealing with a biologic, there is a concern about the different individuals involved. So whether we're talking about animals, and then we're looking at monkeys or rats or mice, and then we have human tissue. So you have the capability of having immunogenicity as a result of that. Things that are more mouse related than human related tend to be less allergenic or immunogenic in the models, but then you have to figure out how to translate that to man. So the whole process of doing biologic development is particularly focused on the whole immunogenicity process and which things can we do, generate useful information versus those things we can't do because we're going to stimulate an immunologic response that will essentially invalidate all the data that we're going to generate. Non-clinical studies continuing along that line. There's cell-based testing, in addition to animal pharmacology and toxicology. And here the key is to define what, and this is true whether you're looking at biologics or whether you're looking at drugs, you need to be able to qualify the information so that you have enough safety, potential efficacy information to proceed to that first human study. So all of that's done at this point in time. In addition, as I mentioned, there are the quality guidelines. You've got biologic guidelines for the active substance and then the drug product. And I'm sure by now you know this, but the substance is the actual chemical compound or the biological compound, and the product has to do with whatever has to be incorporated with that drug substance to make it stable, to make it useful, to make it non-toxic. Other items of particular importance as you do the biologic or biotechnology development have to do with the quality of the manufacturing processes. And keep in mind that since these processes are essentially biologic and or living systems, if you want to look at it that way, a minor change in these kind of processes may actually have a very significant impact on the quality of the product, on the efficacy of the product, and on the safety of the product. So clearly, A, you don't want to be making very many changes, and B, if you do, you have to be very careful about their overall impact in terms of the clinical setting for those particular individual products. Sterilization is an issue with biologic products because you have to worry about what you do to that product so that you don't inactivate it. So autoclaving is by and large not useful for biologic products, or it is, but it completely makes them worthless. So there are different ways of doing that. One way that's commonly done is membrane filtration. So you progressively move series through tighter and tighter pores to get rid of unwanted materials. So we've gone through some of these kinds of things. As is true for drugs, you need to do lot production and then stability testing on those lots to make sure that you have stability that is in a longer duration than whatever you're planning to use in the clinic at that point in time. So if your first study is going to be a week, well, you need to have stability that's longer than, if it's going to be 3 months, again, you have to have stability that's longer than the amount that you're going to be using in the clinical trial. Again, those kind of comments really are true for drugs or biologic, but it gives you a sense of what you have to do with the biologic. We talked with the graph a little earlier about the starting material being the master cell bank. From there, you need to know where all the cells came from. And then from that master cell bank, as you take sample cell banks and create secondary cell banks, you need to have a map that essentially refers back to the original cells so that whoever is developing this can trace it back. If you're the regulator, you can follow it back to wherever it was initially started. One of the key things in biologic products is the testing for adventitious agents, in particular for testing for transmissible fungiform encephalopathy. I get the word out. TSE, very important in Europe. a source of contention between Europe and the United States several years ago, but it has remained as a testing requirement for biologic products. And so you need to just build it into the list of things you have to do. Source of materials is also very important. What you're trying to do, particularly in the biologic side, is to avoid animal-derived materials, and because they can contain viruses or oncogenic agents within them. You also will need to come up with functional assays. So as we get to biosimilars, we refer back to these kind of functional assays because they're critical for characterizing the product, the biosimilar, in terms of how it looks back at the reference medicinal product. Obviously, GMPs, one of the concerns, this is true, again, whether it's a drug or a biologic, is that there's a bridge between the lots that are used in the non-clinical studies and the clinical studies, and within the clinical studies, so that a lot used early is the same as a lot used later. On the clinical side in the drug world, we tend to start with smaller lots and then to expand those lots as we move through the course of development. In biologics, that is also true in terms of how we tend to progress the development, but we tend to move to larger lots earlier so that the manufacturing process is stabilized by the time we're getting to phase 3 clinical trial. But you still need to bridge the information between the lots to make sure that lot 2 is the same as lot 1, 3 is the same as 2 and 1, etc. And here the wild card is the fact that they're biologic processes, so it's a little harder to keep them stable than it is for the new chemical entity type of manufacturing process. Clinical studies have to be tailor-made for each product, so keeping in mind this biologic, depending on whether it's an antibody, whether it's some other version of a biologic product, we have to figure out the intended population, the route of administration. Now, again, these are true for drugs also, but really a key consideration as you look at the biologic, because again, there's the possibility for immunologic responses that can occur in patients who receive this particular product. There are a set of guidelines that are produced, published on the EMA website by the CHMP that at this point cover a pretty broad range of therapeutic areas. And it's true for biologics as well as it is for drugs in terms of the applicability to those areas. But they do make special mention of biologics for each of those therapeutic areas. So if you have a therapeutic area you're working in, as opposed to worrying about the test for the moment, then it's useful to look up those guidelines by therapeutic area and try to see the applicable guidances that are provided within the documents for drugs and or biologics. One other piece of useful information, which you probably already know about, are documents called EPARs. Those EPARs are released at the time of approval for all medicinal products, so whether it's a drug or a biologic. But they're particularly important for the biotechnology products because they do provide a fair amount of information about manufacturing processes, quality controls for drug substance, drug product, as well as, particularly as we get to biosimilars, those kinds of things that have to do with the qualification and the release of the product, so that as you're developing the biosimilar, your product can meet those same sets of criteria in terms of release, stability, overall efficacy. So the EPARs are very useful. I provided the website there to make it easier for you to track them down. And again, drug and biologics are located in that same section on the EMA websites. As you go through clinical development, One of the things that's absolutely crucial is that whether you're looking at phase 1 studies, phase 2, phase 3, that you worry about immunogenicity in the sense that you're continuing to monitor it all the way through your program. The regulatory agencies, doesn't matter whether it's EMA, MHRA in the UK, or whether it's the FDA, they are going to care a lot about those immunogenicity data. So it's very important that they be done, and that they be done well, and that the characterization of antibodies, in particular anti-drug antibodies, is done in such a way that the regulators believe the data and understand that you don't have a problem with regard to your drug in the patient population. Size of studies, similar to drugs regardless. So the overall development program looks the same. The one wild card, of course, as we talked about a couple weeks ago, has to do with orphan products. So if you are looking at rare diseases and you have a biologic, you're now confronted with the challenge of developing that biologic in a smaller patient population, but the same sets of rules ultimately apply, meaning you still have to have the product characterized, still have to worry about immunogenicity in the early clinical studies, and the development program, even if it's truncated by virtue of the size of the population, still needs to follow at least the general paradigm of phase 1, phase 2, phase 3, or smaller population followed by a larger population. So the marketing approval application, remember I said that there were different types, and then there are different filing routes. And I'm going to cover this briefly. We talked about it previously as we looked at the orphan section. It's applicable regardless of what you're looking at with regards to what you need to know in real life to move products through the EMA, or what questions might show up on the test. So we'll cover this briefly at a pretty superficial level, but the data that you need to look at are here on the slides, so you'll be able to look at them later. So this is the CTD format, and it identifies where things go. And this format actually is listed on the slide is true other than module 1. This is true regardless of which region you're applying for. So the items that are in here, if you fill those in, that application can go into the FDA as well. Again, module 1's different country by country or region by region. So the types, so we covered the format. Now these are the types. The types of dossiers are either full or abbreviated. And what you can see is the type of application on the left-hand side of the slide. So you've got a full application, so new chemical entity or a new biologic needs the full application. You see all the data that needs to go in there. The legal basis, those they aren't going to, at least in my experience, they're probably not going to show up on the test when they ask you those numbers, but it does give you a sense of where the information is located. If you look at generics, you look at hybrids, biosimilars, you can see the different characteristics or requirements for each of those types of application. And again, if you care, the legal basis over on the right-hand side. So then we've got format, we've got the type of the application. Now we'll cover the route of the application or the route of the filing. And basically it's for centralized, the mutual recognition. Mutual recognition means it's already approved at least in one country, and you're now asking country 2, 3, 4, et cetera, to refer back to the first one and to recognize that that one applies to your country as well. The decentralized says, well, I really do want to apply for more countries, but I really don't want to do the whole centralized. So it's a way of doing that. You have a reporting country, co-rapporteurs for that particular application. And then if you really only want to sell your compound or your product in one country, well, then you've got the national procedure available for that. So a little bit more detail for each of these particular. Again, as I mentioned, there's always a concern about the legal basis. So you'll see those items on the right-hand side of the slide. Keep in mind the centralized procedure, whether you're required to do it because of the type of the project, or you choose to do it because you think your product has special characteristics, deserves to be approved via the centralized procedure. Single application, single review, single approval, and you can now sell your product in all 27 member states. And for most of these, that also includes the countries that are part of the EEA as well. Skipping through the rest of this, other pathways, we talked about these 2 weeks ago in terms of orphan, the accelerated assessment, which really gets you an opinion in 150 days. Remember that magic number for orphan, less than or equal to 5 in 10,000. That's the prevalence. In addition, conditional approval can happen that's ultimately converted to a standard marketing application, or those that fit into the category of exceptional circumstances. Again, we talked about this 2 weeks ago, but they're here just as reminders. In addition, there are a couple other pathways, one called Prime. I provided the reference for the website listing for you. This was to, again, enhance the development of medicines that target unmet medical need. This offers extra support to optimize the data generation and does qualify for accelerated assessment. It does use a centralized procedure. There is the option for compassionate use. There's also an option for hospital exemption, meaning the product's not going to be sold any other place, but could be used in a single institution. Sometimes I've seen it in a couple institutions, but it's not widely used. And it tends to be focused entirely in this group of products called advanced therapy medicinal products. Registration, I'm not going to cover a whole lot of this detail because I know it's been covered in other lectures, but just quickly as a reminder, must be used for products developed through biotechnology processes, including biosimilar products, if they've been approved this way. You can choose to do a biosimilar that's not gone through a biotechnology process, for example, an earlier product, but there still is some advantage to going through the centralized procedure. The legal basis, now this is a regulation and it covers a number of different items in there. We talked about the common technical document format. Also, I mentioned the TSE, also TSE, BSE are both versions of this testing that need to be done. Marketing authorization, once it's approved, you have a renewal at 5 years after the initial marketing application. Subsequently, they can extend that so that it's an open-ended approval, or they can require you to come back in 5 years for a second review. In either case, it still turns out to be a good way to go through these processes. So once you have your product on the marketplace, and again, keep in mind, I'm talking about biotechnology products in general, although what I'm going to say is also applicable to biosimilars. So just as a way of kind of framing that portion of the discussion, post-approval changes are called variations in Europe. In addition, you almost always are going to have particularly for biotechnology products and or orphan indication, you're going to have post-marketing surveillance and pharmacovigilance requirements. So with regard to potential variations, there is a comparability guideline, and this is important to also consider as you think about biosimilars, because it's the same logic, meaning that what I made here, I have to be able to demonstrate that the version number 2 is the same as version number 1, or my version is the same as the reference medicinal product. The logic is the same here for this comparability exercise. And it defines that on what you can see here on the slide. So it's physical, chemical, and biological characteristics. I mentioned those up front. The quality, so that is the CMC manufacturing process. Safety, looking at the non-clinical testing, and then of course clinical, both from an efficacy standpoint as also from a safety aspect as well. So keeping in mind that especially for biologic processes, even minor changes may result in very significant modifications that have to do with either the quality of the product or the safety of the product, or importantly, efficacy, or worse, I guess, some combination of those. So post-authorization variations. Regulatory dossier changes can be requested by either the market authorization holder, so that is the sponsor, or the regulatory agency can also say, hey, we think there are some issues here in terms of documents, so we want you to make the following changes. So as you look at the categories over here, you've got 1A, IN, 1A, 1B, and 2. And hopefully I tried to keep the descriptions as narrow as possible so that it's easier to remember what these things look like. So the definition of the change is really there in terms of the underlying part. It's well-defined, but it's a very minor change. You can do it and then tell them about it later. Same thing's true with the 1A. So in this case, you can implement them immediately, update it in the dossier eventually. In fact, you can group a whole batch of them together in that periodic report. Again, fits into the category of do it and then tell them about it later. Once you get to 1B, now there is a question as to whether or not those changes might actually have patient risk associated with it. And the patient risk, again, could be either safety or it could be efficacy, either way. But in this case, you now submit, so that's the tell. You have to wait the 30 days. That's the wait part, assuming you haven't heard anything, or if they've already provided the information. Once you've done that, then you can actually go ahead with the changes. Major changes require lots of documentation. They require new benefit risk evaluations. And before you go through the process of actually implementing them for the marketed product, you have to have formal regulatory approval. So that's in Europe, that's the set of variations. Those are true whether it's a drug or a biologic, but in any case, here's what fits into the category of authorizations. Pharmacovigilance, again, this is true regardless of whether or not we're talking about a drug or a biologic. We now have to do post-approval safety updates, or PSUR. It really means that on an annual basis, which is looked at as this data lock point, we resubmit all the information that updates the dossier from the time that we submitted to now a year later, and then on an annual basis from last year to this year at each interval from that point forward. And in Europe, you can now do these via electronic submission. So that's some of the information that there in the bottom of the slide. If there are changes that actually seem to have greater impact, those fit into this category of urgent safety restrictions and can affect the summary of product characteristic items. So that's basically the product label. If it changes that, then those are items that really need to have formal review, have formal approval prior to moving forward with those. post-approval referral. Now, this is a situation in which either the agency or a state or the marketing authorization holder can say, oh, we need to resolve some issues. And so therefore we're going to do the following thing. If you're the marketing authorization holder, you're going to have this in mind. You're going to put in place various systems in order to make sure that you've actually affected or effected the changes that you need to have in place for any one of the items that are mentioned there. On the other hand, you'd really rather not have either the EMA or the European Commission as sort of the surrogate for the EMA, or the Council for Medicinal Drugs, or any individual member state initiate this post-approval referral process, because that means you're now going to play catch up to try to answer what objections are identified. So this slide summarizes essentially the key aspects of that particular process for you as we're trying to put that into perspective. A couple potential exam questions here related to these things. Manufacturer wants to gain approval of an over-the-counter product. We didn't really talk about this, but the logic here is the same. We'll get to OTC products later in the course. And the product's already been approved in a reference member state by another competitor. Manufacturers should file a type 2 variation, type 1, forget it, it's an OTC product, or you need to file a new marketing authorization application. The answer is file a new application. There's an explanation for you to justify that particular answer. Another question, according to EU legislation, companies may advertise to the general public all drugs, non-prescription drugs, medicinal products on a medical prescription, if cleared by the advertising committee, medicinal products that contain psychotropic or narcotic substances. This is actually a real question, but should be pretty straightforward for you. It really can only advertise non-prescription drugs in the European Union market. Number 3, this is, and again, we haven't really talked about generics yet, but to get approved as a generic, company submits to the authorities accompany the originator's market product approval dossier. You probably can't get your hands on that unless you are, and I'm going to pick Merck, and you now want to have your own generic. So yeah, you can get your hands on that particular application. But if you're Ralph's new biotechnology company, you're not going to be able to get your hands on that original Merck application. Or you do CTD modules 1, 2, and 3, and you prove that the generic is bioequivalent. You've got some kind of data exclusivity contract with the originator, or you simply file a manufacturing quality module. Even though we haven't talked about the generics yet, this should, again, be relatively straightforward for you. So you're really filing an abbreviated application. Remember those things we talked about earlier about the types of application. So generic fits into the category of being abbreviated, and it only contains CTD modules 1, 2, and 3. The 1, remember, is the one for the region. So it's really only 2 and 3 at that point. And that is it for the first part. So I think what we can do, I realize we're about 10 minutes early, but let's take a break. And then from here, we'll come back and we'll do the biosimilar talk. And hopefully that will work out for everybody. So how about 5 minutes if we come back at what your time will be 6:55. And
Perfect.
there you go.
Thank you, Barney. Okay, everybody, welcome back. It's 6:55. We will go ahead and get started. Marty, are you ready?
I am ready. Okay, can everybody see the slides?
It is showing very small on a very wide screen.
Better?
Now we just see your desktop. I really like your background.
Okay, well, let's start over and see how we do. It all works so well. And at the beginning.
It was too smooth earlier.
Way too easy, yeah. Get rid of that guy and I go here. And that is good. That. And we go there. We'll pick this. And it's lucky we only got that there. And no. How are we doing? Okay. All right. So from here, we will go to biosimilar products. We will cover the European Union and we'll cover the international / rest of the world, which is now a separate portion of the book. The way this is organized is really to give you a single picture. So rather than going through the European Union and then going back through international, I've grouped them all together. Much as we did for the last lecture 2 weeks ago, we'll try to identify those common elements that are true more or less regardless of what area you're in. And then we'll move to the specifics for each of the individual countries or regions from there. So this is how we'll cover it. We'll again start with the overview and then move through the specific regions, ending with Africa and the Middle East at the end. 40,000-foot view here. In the 1980s, the first recombinant DNA products were approved by both the European Union and the FDA. Those products, supposedly the easy, I guess, if you want to look at it that way, products were insulin and somatotropin, which is a growth hormone. Then it took up into 2005 before the first biosimilar product was approved, in this case by the EMA. And the EMA really moved ahead of the US in terms of this whole process early on. Remember, as we talked about orphan products, it was really the reverse. You know, the US really took the lead on that, and it took a while for other countries to catch up to us from that standpoint. Here it's the reverse. The EMA really pioneered the whole process of looking at biosimilar product and giving structure for the review of those products, which they put in place and then began to approve those products over a number of years. So much as this, we talked about with the orphan products, there are different definitions of what qualify as biosimilar products. Fundamentally, and you can look at all the different terms and the definitions that are applied here, but fundamentally what it is you have a reference product, in this case, a biotherapeutic product, and we've now created a new biotherapeutic product, which is similar to that reference product in terms of quality, safety, and efficacy. That's really the key thing, regardless of which definition you want to look at as you go down this particular slide. That's the fundamental relationship that we're looking at with regard to biosimilar products. So again, the commonalities I mentioned the comparability exercise early on, and I mentioned it in terms of really looking at, say, lot number 1, lot number 2, lot number 3. Here, we're looking at our product in comparison to a reference biological product, or you want to look at a different term, it's the biosimilar medicinal product compared to a reference medicinal product. However you want to look at those terms, the first thing is this comparability exercise. We'll talk a little bit more about this later. In addition, we need to say that the product we're talking about, the biosimilar, is similar in quality characteristics, so chemistry, manufacturing, control, the biologic activity, those are the assays that we're using to try to determine its biologic potency, and then in the clinic, its safety and efficacy. And keeping in mind that it's a biosimilar product, so there might be some minor differences in terms of inactive components in this product. So, comparability exercise, it's similar, and it's similar to an already licensed reference biologic medicine or reference biologic product. And at the end, the regulator is going to be able to say, there are no clinically meaningful differences between the biologic product that we've got, and we're trying to move forward, and the reference product in terms of safety, purity, and potency. So those are the really key things. And as I said, if you get that and they get the components to go underneath each of them, that's pretty much the same regardless of what the geography is. You've seen this slide before. We talked about it in terms of looking at biologic products in general. It's the ICH scheme. Although if you look on the websites in the EMA, you'll see exactly the same diagram. And it covers the non-clinical elements, quality, as well as the non-clinical elements of functional activity, looking at the safety, the toxicity studies. You've got the specifications list, which really has to do with that set of physicochemical characteristics, which enable you to say, yep, this one's the same as that one over there. And then, of course, you're demonstrating that the clinical efficacy and safety look essentially similar. So with the biosimilars in general, the items circled in red here are the ones that are really crucial in terms of saying that that reference medicinal product and our product are the same, in this case, similar, and therefore we can now put this product forward. So again, that set of comparability exercises, the critical quality attributes, and then demonstration that it's similar in the clinic. In general, if you're trying to apply for approval, this is what the information looks like that goes into the summary. The format, pretty much everywhere at this point in time, is the Common Technical Documents format. Reference product requirements. Most of the time, it requires that the area in which you are applying for approval has also approved that other product. If you're in some of the smaller markets, they may say, well, and we'll talk about some of the details later, they may say, well, this product's approved in the United States, it's approved in Europe, and so if you're going to do a biosimilar in our country, just show that it's similar to the one that's already approved in the US or in Europe. So depending on the country, that's a potential pathway. The exclusivity periods for the reference product, for the most part, really only defined for the United States and the EU. And then we talked about this a few slides ago, but really this is the process for establishing biosimilarity. You'll see the word stepwise approach, and that's really the logic that you start with a set of comparability tests or exercise looking in vitro, and then moving through demonstration of similarity for manufacturing processes, that the non-clinical studies looking at safety toxicity are similar, Then you've got the clinical studies that focus on PK and pharmacodynamics. And then you've got the clinical studies. And then at the end of that, you have demonstration that this product is similar to the reference medicinal product. So that when you see the term stepwise, those items listed with the bullets are the ones that really fit into that category of stepwise approach to demonstrate the biosimilarity of this product. In addition, you'll see the quality data requirements. Here again, what you'll see is the references to the Q series of ICH guidelines. From there, you've got the non-clinical data requirements, the in vitro, in vivo studies, clinical data requirements. By and large, you have PK demonstration. You have some evidence of pharmacodynamic similarity, which might be biomarkers or might be easily observed clinical benefits. And then if you get to that point, then you can do longer studies, sometimes depending on the population, or this is sufficient for ultimately submitting the approval application. In addition, another term that will be very important is something called interchangeability. So you can have a biosimilar product, and the pharmacist in Europe and in the United States cannot exchange your product for, cannot use your product in preference for that reference medicinal product if the physician wrote for the original reference medicinal product, unless we've been able to demonstrate interchangeability. In that case, then the physician writes the prescription for the reference product, but the pharmacist can, by law, it depends on which state you're in the United States, can then submit, then can actually deliver the biosimilar product instead of the reference product. So interchangeability is a big deal. It's a big deal from the standpoint of being able to sell your product. It's also a big deal from the standpoint of being able to, within the legal system, have that product substituted for the reference medicinal product that might be stated on that prescription. In addition to that, you'd like to know about, do I have to use only the indications that are in the original product's application and summary of product characteristics, or is there some way that I can expand those? In general, in Europe, at any rate, the similar biologic product will qualify for all of the indications for that reference product, whether you studied them or you didn't. So if you've been able to demonstrate the biosimilarity, then you can acquire all of those indications, even if you didn't actually study them individually. There is, in general, pharmacovigilance requirement for any biosimilar product, and that's true whether it's the United States or not, or Europe. And then labeling, whether you call it the product insert or product summary or the product label in the United States, or a summary of product characteristics, which is the usual term in Europe, the similar product will be either the same or will be a subset of that reference medicinal product. Now, that latter part will depend very much on the geography in which you're applying. And then finally, what about naming? The World Health Organization's tried to impose a standard which includes a set of biologic qualifiers, a letter code with an INN name. That's not quite been agreed worldwide, but it's much closer than it's been in the past few years. So we're making progress on that. The whole point is to have a standard name that is applicable around the world. In terms of agency consultation, review, evaluation, you're applying to the regulatory authority in the country in which you're trying to get this product out. And you're going to have to understand, once you get outside the Europe, you get outside the United States, I think I would probably add the UK to this. There's a stability to the regulatory processes for this type of an application that you can count on, you can plan for, you can anticipate, you can understand. Depending what other geographies you're looking at, there may be some variation, and it might change in the middle of your development program. So you have to keep paying attention to those things. Post-approval requirements. Typically, as the condition for approval, you'll have some kind of safety or monitoring things you have to do. Occasionally, it might be a registry if it's an orphan product. And then, obviously, if you have any change in manufacturing or you hear of changes in safety experiences, then you need to identify those. I mentioned previously in the last portion of the lecture, as we talked about variations as being the terminology in Europe in terms of the changes that happen post-approval. So keep in mind what you see in the red on that paragraph at the bottom, or the box at the bottom there, is it's important to realize that the reference product and the biosimilar are, quote, tied at the hip with regard to safety. So that means that if there's an issue with one, in your case, that could be the reference product, you're stuck with it. Whatever happened there is now applied to your product label. And by the same token, if we have a problem with our product, our biosimilar, that problem will now be transferred to that reference biologic product. So once the biosimilar is approved, then you really have to pay attention to that reference medicinal product as well, in terms of whatever happens with it. So those 4 slides, I don't claim that I invented this format, because I stole it from one of the international books in terms of how they present summaries for individual countries. But if you look at this as being a set of general topics, and then you begin to fill them in for each of the individual countries or regions, it's one good way to compare as you go from one to the next to the next. So as overarching sets of principles, these 4 slides that I've just gone through should give you a good feeling for how you get a biosimilar product into the marketplace and the requirements they're going to need to satisfy in order to get it there. Recapitulate a bit, and there is a fair amount of repetition here that's on purpose. The basic principle underlying the development of the biosimilar product is comparability, not identity. It's comparability with the reference product. So that means the quality, the safety, the efficacy are comparable, and that translates to saying there are no significant differences compared to the innovator or the reference product. So for quality, for biologic products, that means the production and manufacturing processes are the things that really define the product. So as we mentioned previously, if there's a minor change, it might actually have major consequences. So as part of that quality process, you need appropriate assays for stability. You have to understand all the manufacturing processes. So you've got details and you've validated the individual components of those manufacturing processes. You understand the characteristics of the product. So the primary, that is the amino acid sequence, secondary, the linkages, and third, tertiary, the folding scenario. You're looking at 3D structures. They must be comparable. It's not identical, but at least functional groups are showing up in the similar location in this molecule. The pharmacokinetic profile needs to be the same. Again, not exactly the same, but it has to be very close. Sterility, of course, is a key component. We talked about excipients and compatibility assessments. So those are items under the quality title that we have to pay attention to as we're looking at this comparability process. Safety, we have to understand the characteristics of the reference product. Again, we have appropriate assays for stability, as well as ultimately we're going to have the same things for efficacy, for potency. Here again, let's talk about the sequences, maintaining the PK profile, etc. So the types of analyses that we need to pay attention to are ones that are able to show equivalent of product 1 to product 2, or product 2 to product 1, probably more appropriately. And in this case, it's the quality, non-clinical, and clinical. And the point of equivalence has to do with saying that's the approach that's preferred from a statistical and an analytical standpoint than saying product 2 is non-inferior to product 1. So there are different statistical tests which enable you to say one is equivalent versus non-inferior. But the important point for you to consider as you're thinking about tests and answering questions is to understand that clear, as we're looking at comparability exercises, our goal is to have equivalence, not simply non-inferiority. The table below, which comes out of the international book, really identifies the characteristics of equivalence versus non-inferiority. You could argue there are some disadvantages to the equivalence. You do have to have more subjects or more data points. It's probably a better way of saying it, since this logic applies to non-clinical as well as clinical observations. But in any case, that table should give you a pretty good sense of the comparison between the two. So as you think about this stepwise development that we mentioned previously, we're really going from the reference medicine to the biosimilar medicine. And we're going through a series of steps. Step 1 looks at comparability with regard to quality. Here, we're looking at really the analysis of physical and chemical properties, and then whatever its biologic activity is looking like. And those are the potency assays that we talked about a couple moments ago. From there, from quality, we've now moved to non-clinical. So we need to look at the pharmacodynamic effects in whatever models seem to be appropriate and are consistent with prior observations, so that reference medicinal product, as well as making sure that we can define the immunogenicity as best we can within these animal models. Again, it's going to come up as we move down to step 3 in the clinical studies, but we need to understand what the possibilities are here in terms of immunogenicity and have to figure out how we're going to evaluate. Then obviously it has toxicology studies to look at as well. So as you look at step 1, step 2, and then ultimately step 3, the information that's available for the reference medicinal product becomes crucial because, for example, the EPAR in Europe or the summary basis of approval in the United States will identify the studies that were done for that reference medicinal product. So those are your at least core benchmarks to look at as you proceed through this pathway, and then ultimately down into the clinical studies where we're looking at both PK and pharmacodynamic effects, ultimately efficacy, safety, and then, as I said, the immunogenicity characterization in the clinical setting. So if you want a picture that gives you a sense of where you start and where you end, this picture probably is a pretty good way to conceive of the whole biosimilar process. So those are the overview slides. That gives you a sense of the questions to ask for individual countries. So if you look at that grid that we showed a few moments ago, those 4 slides, those will be the key questions that we ask as we proceed through each individual region. How can we fill in that grid for individual countries? And in some cases, there will be boxes that will be empty, or there may be additional boxes depending on which country or which region we're looking at. So we'll now move from the general and head on into the specifics. And first, we'll cover the European Union. Some of this will look very similar since we referenced some of this information in the prior portion on the biotechnology approval. So keep in mind the definition is biological medicinal product contains a version of the active substance of an already authorized original biological medicinal product or reference medicinal product in the EEA. So that's the European Union plus the couple extra countries. Initially, we're looking at the directive that tells you what the requirements are. So as you think about directives, the whole process of biosimilarity and approval of products, the European Union really moved forward based on this set of definitions. And even though we're moving into regulations related to these items, this has been a pretty effective process that the EU has put in place. So what does it say? And fundamentally, it'll be similar to what we've already talked about, but the biosimilar will fulfill all the requirements for Module 3, that is quality, and satisfy technical requirements of the European Pharmacopoeia and relevant guidelines. So this fits into Q5E, the ICH guideline. So remember, this is the first set of technical comparability exercises. We're then able to move through the general technical and product class specific provisions for safety and efficacy. We have an active substance that is the same, in quotation marks, and then we're going to have the same route of administration and the same frequency of administration, that is the pathology. So that's the core set of definitions of what a biosimilar product looks like in the European Union. So if there are deviations from that original reference medicinal product with regard to any of those items listed there, you're going to have to justify. In addition, then you're going to have to say that any differences that are present do not compromise safety. That's going to be the first question. The second would be then, well, what happens to efficacy? Does that change? If any of those items in the bullet points above are different for our product compared to the original, what happens to safety? And then what happens to efficacy as well? So there are a set of guidelines that are identified in Europe, and we mentioned those previously. These are sort of at the top level, the kinds of things you need to consider. We talked about emphasizing the similar products comparability to the reference product. We have to look at the activity in the original reference product, summary of characteristics, their EPAR, and then from there we understand what we have to show in terms of efficacy and safety, and look at the potential for the generation of an adverse event that may require a fairly large number of administration prior to the occurrence of such an AE. Clinical pharmacology studies become a big deal, so to speak, in the sense of being able to demonstrate similar pharmacokinetics, pharmacodynamics, either as a marker, some kind of biomarker, or some kind of clinical effect, benefit, and within that category, we need to consider that immunogenicity is not present as a result for our product compared to the other product. So the other thing to emphasize is that non-inferiority analyses They're acceptable for some studies, but not for efficacy. So keep that in mind when we talked about equivalence. Non-inferiority is not going to get you there. You have to have equivalence on the efficacy side, whereas non-inferiority for safety as an analysis or a set of analyses may be reasonable depending on the product. So how do you get from one to the other at a practical level? Well, you start from the reference medicinal product, and it's this may be a graph or a cartoon, so to speak. It just looks at the same information we've talked about, but maybe looks at it in a different way. So maybe by looking at it this way, it summarizes it for you and fixes it in your head in a way that makes it a little easier to figure out. So first of all, you've got the target definition and characterization. Then, as we've talked about already now multiple times, you move to the quality part of the process. Then you define the comparability from a physical, chemical, and a biological standpoint. You've done comparative non-clinical, then comparative clinical studies. You've put together a risk management plan. Once you've got all that in place and you say, well, what we've got is very similar to what the reference product looks like, we now begin to file our application. If you step back a little bit and try to figure out, well, how long does it take to do this? Keep in mind that it takes anywhere from 10 to 15 years to put together a brand new biologic product. Here it's shorter because we can do a fair number of things in vitro rather than having to move up into the non-clinical, the toxicology types of testing. But you can see here, if you call these different steps, 1, 2, 3, 4, you get through the cell banks, you get through manufacturing, get through the quality processes. It's still taking you 2 to 3 years to get there. Then you've still got the other aspects of comparability testing. So you've still added another 3 to 4 years. So it still takes you a while to get where you want to go, but it's still faster and easier than it is developing that original reference medicinal product. Different way of looking at it, different cartoon, so to speak. So you're characterizing the reference medicinal product. So you've understood what is in the EPAR or in the summary basis of approval. We've now taken all of that information. We've created our own processes for developing our own product. And then we're confirming ultimately the comparability between our biosimilar product and the reference product. And again, turning it on its head in a slightly different way, similar to the graph we had previously, the reference medicinal product on the left, the biosimilar on the right, and you're moving from step 1 to step 2 to step 3, the quality, non-clinical, and clinical studies to get from one spot to the next spot. So in Europe, keeping in mind we are first focusing on Europe, we have the centralized procedure. It must be used for products developed through biotechnology processes. There's the legal basis. I knew you wanted to know that. Format is the common technical document format. We must pay attention to the biotechnology specific guidelines issued by the EMA as well as the ICH. We talked about transmissible and bovine spongiform encephalopathy tests. Those must be included. Again, you've seen this slide because we talked about it previously. If the prior product went through a centralized procedure, then you have to do a biosimilar will have to go through that process. However, if the reference medicinal product was authorized via national procedure, then you have other options. If that was authorized through the centralized procedure, you have access, but you also have a requirement to go through the centralized process. If that product were authorized via national our mutual recognition process is decentralized. You could follow those pathways, or you could request approval through the centralized pathway. Now, some of the criteria here having to do with the centralized pathway and the other requirements get to be a little subtle. So what's on the slide gives you a sense of the potential variation. As a way of thinking about it in general, depending on how a question on a test might be phrased, a centralized procedure must be used for products developed through biotechnology processes, including biosimilar. But had that product been approved a different way, then you have some options that you might be able to use rather than going through the centralized procedure. Now, my process, my way of looking at the world, I tend to prefer the centralized process because it's really well defined. And if the reference product's already gone through this, then I really understand what that roadmap looks like. And so therefore it makes lots of sense for me to continue to follow that. Not over and above the fact that I may be obligated to follow it since the reference product had already gone through there. So this slide hopefully will help you frame that. Keep in mind there are some subtleties there that, depending on the way the question could be worded, it could be set up as a trap in the sense of must or maybe or possibly, depending on what the requirements are. So hopefully that helps as you take a look at this. Well, make the assumption we're going through the centralized procedure. Now what happens?
Bernie,
We submit. Yep.
sorry, real quick. If we go back to the prior slide, if a biosimilar or if the reference listed drug went through a previous, a different procedure. So let's say it went through the national procedure. How difficult is it for EMA to get access to that information if you wanted to go through the centralized procedure?
It's actually not very difficult at all.
Okay.
In fact, it's surprisingly not difficult in the sense that all of the regulatory agencies in Europe, and it actually includes the UK, the US, and Japan. If you file in one place, those applications are visible to regulators in other countries.
Okay.
So it's actually easy for them to access that information. Now,
Okay, so
there could be challenges depending on the age of the prior application. But if we're looking at biologic products, they're all going to be fairly, quote, modern. And so the information should be available. Ones that are particularly new chemical entities that were approved in the, say, late 1990s or early 2000s, some of that may not be quite as easily accessible. But for this, yes, should be.
Do you have an idea of how many biosimilars are approved right now in Europe?
I do, and there will be a list.
Okay.
So just hang on.
Is it broken down too by procedure?
No, it's not. The ones, and not only that, I do give you the reference so you can go track it down and look at it. And the advantage to both the US and to Europe is that by going into the databases, you can actually cut and slice by, say, for example, indication or countries, etc.
Okay.
So it's actually pretty useful.
Thank you.
So one of the other considerations is, say, for example, just apropos of the question, the reference medicinal product was approved, say, a decentralized pathway, and you want to go through the centralized pathway, you will include a justification for the eligibility. Now, what that fundamentally means is, hey, listen, this product I've got is for still a pretty unmet need. I'm going to be able to offer it at a lower price with the same quality and same safety characteristics. So that should be enough for you, EMA/EU. And in my experience, it is. So they have a preference actually for moving things to the centralized procedure. Might seem as though they might be trying to get rid of things to cut down on the workload, but actually they've been very good and very efficient. COVID was a challenge, but it's getting closer to normal at this point. One of the things, of course, which is true for these types of products as well as is for generic products in general, has to do with the protection periods that have been applied, data exclusivity, marketing exclusivity periods attached to the primary product. So whether it's a new chemical entity or whether it's a biologic product at the time of approval, they will have those exclusivity perks built into the approval process. So we have to pay attention to those before trying to submit the product. If we submit an application prior to the time that either the data exclusivity, that one's really a bad one if you try to go past that one. But say, for example, marketing exclusivity, or there are concerns about patent life, and you submit, your application will be returned because those exclusivity periods will not have expired. That's true in the United States as well, just a matter of interest. In general, for biosimilars, you will notify the agency about a letter of intent to submit. This has become less of a requirement, but I, even at this point in time, would say it makes sense because what it does is if you submit that letter, it triggers the appointment of the rapporteur and the co-rapporteur based on the application that you've got, as well as the reference back to this reference medicinal product. So once that's in place, then these rapporteurs are appointed and you actually begin the process even though the clock per se has not started. So I still think it's a useful piece of information. The pre-authorization procedural advice document was updated about a year or so ago, and you can see the reference down there at the bottom of that particular slide. So if you're interested, you can take a look there. The core information is here on the slide. This is what the data package looks like fundamentally. So keep in mind the things that we paid attention to previously, so quality, non-clinical, clinical data. And then also the pharmacovigilance becomes important because this is really referring back to the reference medicinal product. So you're really surveying all the prior experience with that particular product. Because remember that phrase that I used earlier about being tied at the hip. What they have, we have. What we have, they have in terms of safety experiences. So we need to fully understand that ahead of time. We understand whatever data we've generated with our particular product. And then we put all that together and we come up with a risk management plan, which has to be included with the marketing application in the EU. So that's the core set of information that goes into that data package. So now, going back to that grid that I used previously, again, I fully acknowledge that I stole it. So, but just keep in mind that I think it still is a useful organizing principle as you go through these. So, earlier we showed you what I would call a generalized scheme. Here are the specifics for the EU. So the format is still the CTD, the reference product requirements. In this case, it has to have been authorized in the EU. Now it said must be, but you'll see in parentheses, it says that it's possible that a product is not approved in the EU and you can still use it as a reference. There is an instance of it, but that's not typically the pathway you're going to go. Exclusivity period for the reference product. We haven't really gotten to generics yet, but fundamentally, this is the magic here. It's 8 plus 2 plus 1. So you have data exclusivity period, marketing exclusivity period, and then if you apply for a second significant indication, you get all of a year for that. So you spend all the time working up here to get your primary product in the market, and then you say, well, this was really great for rheumatoid arthritis. I'm going to add diabetes. Well, for all of the work it's going to take you to get that new indication, you get all of one whole year. But in any case, you get something. Remember the process. The fundamental principle is the stepwise approach. So we're starting with the physicochemical biological characterization. We move through non-clinical and then to clinical. The quality requirements. This list is pretty much the core list, whether you're looking at the US or the EU. Non-clinical data requirements are pretty straightforward, and they're listed there, as well as the clinical data requirement. Interchangeability not available across Europe. There can be some individual national authority decisions that enable interchangeability, and in that sense, it's somewhat similar to the United States. While the FDA can designate a product as being interchangeable by virtue of the way our laws are set up in the United States, that does not mean that every state will look at that product as being interchangeable. It depends on the particular pharmacy laws within any individual state. So that's the similar thing here. A national authority could override this interchangeable definition and say, yes, within, I'm just going to pick it, Italy, yes, you can write for the reference medicinal product, and the pharmacist may be able to substitute your individual product at the time of that prescription. The extrapolation of indications, typically it's going to say what has been studied clinically, but remember, you're going to end up with a similar summary of product characteristics. So it says only what's studied clinically, but by and large in Europe, and this is true on the generic side, generic drug side as well, you'll get whatever indications are in that reference medicinal product label as well. The pharmacovigilance requirements are there. Labeling, as I said, you get the summary of product characteristics of the reference medicinal product. Naming still may be a challenge. In Europe, just because you get a singular approval doesn't mean you get a single name all the way through Europe. So their individual countries can force you to have 2 or 3 different names depending on what the product actually looks like. Post-approval requirements, we talked about variations. And again, I'm reiterating that phrase tied at the hip down there with regard to safety. And so that's the quick trip through the Europe. And next we'll move on to the United States. So the basics, it sounds pretty similar. The biologic product and the reference product must use the same mechanisms of action for the treatment of the conditions that we're prescribing it for. And now that's true as long as you know or have a pretty good idea of why the reference product works. Obviously, if you don't know, then it's going to be a bit of a challenge to try to come up with that unless you've generated some new additional information. So that's the mechanism of action. The condition is the same target as the reference product. And then to show the biosimilarity, it's the same logic of the stepwise process. You start with the analytical studies, then you move to the animal studies, finally clinical studies. depending on what the product is, and if you think you have a good argument, you can go talk to the FDA and say, hey, I don't think I need to do X. And if you have a good argument, you may be able to get around that. I'm just saying it's a possibility. I'm not sure if that's going to show up as a test question, but in real life, yes, it is possible that the FDA could waive some of those characters. So, for example, this actually would be a reasonable question to ask the agency. That original reference medicinal product was released 8 years ago, 10 years ago. Since then, we, the community, or we, the company, have actually come up with some analytical assays that are better in terms of showing correlation of, say, efficacy or components, etc. And so we think if we do our assays and they are more efficient, they're better, we get better readouts, maybe we don't have to do as many as the original reference medicinal product did at the time of their approval. That would be a perfectly reasonable question to ask the FDA, and there'd be a high likelihood that they would agree to that, making the assumption that what we think is new and different and better has some kind of correlation to the data that were originally supplied by that reference product way back when. Biosimilars. So 505(b)(2) biologics were originally, they were approved under an NDA. Now the FDA can accept 505(b)(2). We're going to come down to some other options here, but this is a process for having a biosimilar approval. And I'm going to come back, I'm going to skip past this because I think the real information is here. The Biologics Price Competition Innovation Act, DPCIA, part of the Affordable Care Act in 2010, added a section to the Public Health Service Act. Keep in mind that biologics in the United States are approved via the Public Health Service Act, even though it's still run by the FDA. It's covered under different sections, and typical biologics are licensed under what's called Section 351A. Biosimilars are covered under 351K. So the K becomes the equivalent legislation for, say, a generic or what we just talked about, a 505(b)(2) pathway. But in this case, this is, I'm going to call it the more traditional way of getting a biosimilar approved is through the 351K. In this way of looking at the legislation, the biosimilar is defined as highly similar to the reference product, understanding that there may be minor differences in the inactive components, and there are no clinically meaningful differences between our product and the reference product in terms of safety, purity, and potency. Keep in mind that if you're looking at an ANDA for a generic, everything must be the same. So here there's some latitude, understanding it's a biologic process. So to meet the standard of interchangeability, and remember we talked about this a few moments ago as we talked about it in the UK. In this way, our biosimilar product is expected to produce the same clinical result as the reference product in what's called any given patient. We'll talk about how you show that in a few minutes. For a biologic product that's administered more than once, what's the risk of switching back and forth between our product and the reference product? Well, if we want it to be interchangeable, it has to be no different than the risk of keeping that patient on the reference product alone. So you can see the logic here is, well, if at baseline I'm using the reference medicinal product, in this case, the biologic, and now I want to switch to a biosimilar, our biosimilar, what we're fundamentally going to say is the safety experience for just staying on that reference product has to be the same as, say, taking our product for a period of time, then using the reference product for a period of time, then switching back to our product, and then maybe even doing another switch back. So that safety profile for the second part should be the same as the first part. That fits into this definition of interchangeability. And so again, what that means is it may be substituted for the reference biologic product by the pharmacist without having to call up the healthcare provider, the doctor, et cetera, and saying, hey, I want to switch from one to the other. But as I mentioned earlier, the proviso is that different states have different laws enabling that interchange to actually happen. So even though it's covered at this federal approval level, states may have the opportunity to override that if that's the way it's built into the state pharmacy law. So if the FDA approves it through a 351 pathway, you may receive basically all the labeling, or some you may choose to not have all of the labeling, or indications, that is, or you may get all of them, depending on how you want to approach that. So fundamentally, in the United States, what's on this slide really becomes the key way for the product to get approved as a biosimilar compared to the reference medicinal product. And keep in mind that definition of interchangeability down at the bottom. So if the product's a biosimilar, the physician has to write for the biosimilar product in order for it to be dispensed. If it's interchangeable, the physician could write for the reference product, and we could have the pharmacist substitute our product for it. So interchangeability. Again, it's not required. We don't have to have it for the biosimilar, but there are clearly advantages to having that designation. And this really recapitulates some of the information on the prior slide. So I described a few moments ago a way of doing an experiment, clinical trial, in order to try to demonstrate interchangeability. Well, the FDA has been nice enough to provide us a guidance for that, and that's listed at the bottom of the slide. It's only a couple of years old, so it's pretty current. And fundamentally, what you see in the slide is the way this pathway would go. So you've got, on one hand, you may have the reference product, and then you have the interchangeable product. And you can see you've got one group at the top there that says non-switching arms. So that's that reference product. They stay on that. The bottom arm, the switching arm, as it's called, has the proposed interchangeable product. We treat with that. We switch over to the reference product. We may now have some washout period. We might do some PK at that point. We may bring them back for the switching arm. So this diagram gives you a sense of the requirements needed. These don't have to be huge studies, but they do have to be large enough to take into account individual variability in terms of clinical responses. So it gives you a sense of what the interchangeability would look like in the United States. The 351K approval route looks like this. And I'm saying you suggest a pre-IND meeting. The legislation gives you the opportunity for some free meetings along the way. Then this is what that set of what an application would look like in the United States for a 351K approval process. Then these are the key studies. So you've got an analytic goal. So that's the set of assays that you do. Then you've got clinical goals. in terms of what the overall clinical outcome, both efficacy and safety, look like. These are the first 2 approvals in the United States. Just that's really for information alone. There are exclusivity periods built into this, and I've identified those. There are still laws, and I mentioned the state laws with regard to prescription requirements. I'm going to skip through this because this is information that some of which I've covered, but again, I think it's useful from a reference standpoint to compare the different geographies. So as we're getting close to the end here, I'm going to really flip through the slides, unfortunately, but I think the fundamental principles that we've outlined partly as the sort of overall overview at the beginning, and then we've now covered the EU and the US. And if you work your way through the rest of the slides and look at the different geographies, you'll see what the similarities are, and no pun intended in terms of the biosimilars. And then you'll see what the differences are from country to country. And I've provided those grids for as many countries as I can provide enough information for. So as I said, you've got Japan, and again, you've got the grid, then you've got India. India is beginning to change their processes. And at this point, they really have more biosimilar approvals than any other country at this stage. And then in India, then you go to China, South Korea, and then there are comparisons here from different requirements. You've got Mexico toward the end and Latin America. Really not, you can see the huge movement forward in the Latin American countries if you can stick them all on one slide. Asia Pacific, so now you're looking at Malaysia, Australia, New Zealand, Africa. Some of those countries are listed here at the end. So that's what the list looked like. I mentioned that there were some, I mentioned that there are lists of the biosimilars, and it appears that I actually dropped that slide out. So I will provide that, and that information will show up subsequent to the lectures. So covered a lot of material in there. Hopefully the slides are arranged in such a way that you can use them as a reference. And I think in particular for the international, hopefully what we've organized is maybe a little easier to use and or read than what's in the book. Although I think this edition of the book is definitely better than the prior edition in terms of the international. But hopefully that'll give you a framework to work from as you prepare for your test. And hopefully it's useful as you look at real life as well. And with that, I will stop.
Thank you so much, Barney. This has been a wealth of information, and you presented it very well. So thank you.
Thank you. Any questions?
I did have a question about India. I know you were flipping through that quickly. You pointed out that there were more biosimilar approvals there than any other country. Is the regulatory requirements lower there? Is that why there are more, or what's really driving that?
It's driving. What's driving it is that the Indians have been very good at copying other products.
Okay.
And so early on, CDisco put a process in place to enable the development of biosimilars. So it's very similar actually to generics in India.
Okay.
Because of the way the manufacturing is set up and their reverse engineering of pharmaceutical products. That's not really lower standard per se. It's just that, I guess in a sense, it's an economic driver of the country. So let's see what we can do to keep it going.
What would you say is the most challenging place to get a biosimilar approval right now?
I would say that fundamentally it's anything that's off the main list. So
Okay.
if you've got the US, Europe, the UK, I didn't really discuss the UK, but today it still looks a lot like EMA, although obviously you have a different regulatory agency than you used to.
Mm-hmm.
So again, US, EU, UK, Japan. Once you go outside that, it becomes a little more challenging. Even places like Australia, New Zealand, you would think they'd be really similar to the UK, but or the EU, but they've carved out their own pathway. And Canada kind of sits out there in a strange place that they're not even sure they believe there is such a thing as a biosimilar. And they have a very strange term for the product. Anyway,
Interesting.
so I think the core countries, it's within the limits of coming up with any biosimilar, they're much easier. If you want one approved in Brazil, since it's still up on the screen, make sure that you include a large number of non-sequential, small denominational bills. I'm sorry, I didn't mean to say that, but my experiences in Brazil have been quite varied over the years.
I do not have experience in Brazil, but I know from the drug side, at least, I mean, they're going through a lot of changes. So
anyway, so on my to-do list is the place where you can track down all of the list of the biosimilars. The book goes through long, detailed lists, which I think are nice, but they just occupy pages in the books. So I think it's better just have the location where you can find them and then look to your heart's content.
well, thank you so much, Barney. I don't see any further questions. Just a reminder to everybody, in the chat box, you'll find the proof of attendance form and the feedback form. So please take a look at those, and thank you everybody for attending today. And thank you so much, Barney, for your time and expertise. This has been really great.
Could I ask a general question about biologics?
Sure.
If a product gets approved or gets reclassified as a biologic, but it's what would be the best process to? I mean, would there be a process to before the product is required to get a biologics license? Would they be required to comply with biologic requirements, or they would still be under NDA CDER requirements?
Well, when you say it's reclassified, why is it being reclassified?
Yeah, I think in 2020 there were several products were reclassified as biologics because they were really they were APIs. They were FDA took a look at it and they said, well, they should have been under.
Yeah. Okay. So yes, the answer is that you would still have to demonstrate that the biologic is manufactured according to biologic standards, which equals having a BLA. Now they, by virtue of having had a prior, if the product's already had a prior approval, then you can move some of that information over, but you're still with a high degree of certainty going to end up with a new BLA inspection.
And the BLA inspection would come from a different body, right?
Yeah.
And
it'll come out of CDER. Yeah.
And okay. And as part of that, if if they have any observations in terms of meeting different requirements or needing to meet their changing requirements. Have you had any experience with how they classify those?
The quick answer is no, I have not.
Mhm.
On the other hand, I have worked with somebody who has done that. And what she did was to, so there were a couple of the requirements vis-a-vis CEDAR/BLA that that the company thought they had really satisfied, or at least demonstrated that they didn't really need to do it. So what they did is they filed a petition, in response to the observations. And if I remember correctly, there was one requirement that CEDAR maintained, but the other ones, then again, if I remember correctly, I think there were three other things. So three, they were able to reverse the observation, and so that the original application was accepted for those. For one other one, assuming my memory is correct, they had to go back and rework that particular process. But it turned out at the end that it wasn't terribly onerous. Now that's an example, not necessarily applicable across everything, but at least they were able to obviate to redo everything.
Oh, thank you very much.
Sure.
I have a question. This is Nandita. So this is in regards with the small molecules we do it is comparable to the RLD, but for biologic, like a proposed formulation, how do we demonstrate like the biosimilar is relevant to the BLA submission? Like how do we submit it? How do we show it is equivalent to the reference listed drug?
Are we talking about new standard new chemical entity, or are we talking biologic? If you're looking at standard new chemical entity, you're now looking at a generic for that. But that's actually pretty straightforward.
A biosimilar. I'm looking at a biosimilar.
Yeah, with the biosimilar, it's the information that's in here. You really are looking at the individual tests that define the particularly the potency and the physicochemical characteristics of the biosimilar compared to the reference medicinal product. Then you go through the non-clinical studies, make sure toxicity profiles look the same. Then you've got ultimately the clinical assessments, which are really pharmacokinetic, pharmacodynamic in the disease. And then if you're looking for interchangeability, then you have to go through that clinical study that I showed briefly on one of the earlier slides. So that's the pathway for biosimilar.
So it should not be, it's not possible just like a normal control correspondent, how do you look for a small molecule? So we have to process all through this way for the biosimilar product too.
Yes, and the rationale for that is that biosimilars are manufactured via a biotechnology process, which is by definition a living process. So there will be variability. Whereas if you have a new chemical entity, you should be able to nail that compound and its characteristics pretty straightforwardly. But with the biosimilar, that's not necessarily the same. But again, the advantage of the summary basis of approval in the U.S. or the EPAR in Europe is that those documents will identify what the reference medicinal product showed at the time of its application and approval, and that becomes your roadmap. It's essentially the cookbook for what you need to do.
Thank you, Bunny.
Okay. So with that, I'll say I'll see y'all in about a month.
Thank you so much, Barney. Appreciate your
Wish you a
time.
great month.
Thank you, Barney.
Here.
Thank you.
Bye, everybody. Have a good evening.
Bye.
Bye.